Friends, welcome to Biology Made Easy. We have discussed earlier the theories of Lamarckism, then Darwinism, and also we have discussed that of the mutation theory by De Vries. But after the theory of Charles Darwin and the Hugo De Vries theory of mutation, there is a lot of controversy which is more correct. By this time, there is advancement in the science of genetics, molecular science, paleontology, ecology, as well as ethology. Ethology means the study of behavior. So, with the advancement of all these studies, Charles Darwin's theory of natural selection was modified, and that modified form of Darwin's theory is known as the modern synthetic theory or NEO-DARWINISM that we'll discuss today in this video. So the modern synthetic theory actually revolves around two different theories. One is that of the Darwin and the other is the mutation theory of De Vries. But the contribution for this modern theory comes from several scientists like Dubzensky, then Sewell Wright, Stebbins, Halden, J.B. Halden, Fisher, and Mayer. So these scientists, they have contributed to this modern synthetic theory, and ultimately Stebbins wrote the entire theory in the form of a book, and that book is known as the organic evolution, or you can write the process of organic evolution. This is the name of the book written by Stebbins. So the modern theory is also known as Neo-Darwinism. Now, what is the basic difference between the theory of Charles Darwin and this modern theory or Neo-Darwinism? That we must know first, then we will proceed to the theory. So, if we will discuss the difference between Darwinism and Neo-Darwinism, or that is the modern theory. What is the basic difference? Now, the first difference is, according to Darwinism, that is the useful variations. The useful variations are inherited. This was his view. But according to Neo-Darwinism, the genetic variations are inherited. This is one of the difference. Now the second difference is Darwin didn't give any reason why the variations are taking place. But in case of Neo-Darwinism, some reasons were given why these variations are taking place. So no reason for variations. But here the reasons for variations were given. Now another very important difference is, according to Darwin, the unit of evolution is an individual, but according to Neo-Darwinism, the population is the unit of individual. This is very, very important. So unit of evolution is individual according to Darwin, but according to this modern theory, that the population is the unit of evolution. So this is very important. Then another point, according to Darwin, the natural selection takes place through survival of the fittest. Natural selection through survival of the fittest. But according to Neo-Darwinism, it is due to differential reproduction of the population. Differential reproduction. This is also one of the major differences. Now, another difference is Darwin didn't give any importance to isolation, which is a major cause of evolution. But in Neo-Darwinism, that isolation plays a very prime role. No importance on isolation, and here isolation plays a prime role in evolution. So these are the basic differences between Darwinism and Neo-Darwinism. Now you have some idea. Now let us discuss in detail the theory of modern synthetic theory or Neo-Darwinism. So the modern synthetic theory or Neo-Darwinism, we can discuss it in a different steps. The number one is that is the genetic variation. Then number two, natural selection. Then number three is reproductive isolation. And the next is speciation. That means the modern synthetic theory, it can be discussed. The first one is genetic variation. Then the second point is natural selection. The third point, reproductive isolation. And the fourth point is speciation. So we will discuss the modern synthetic theory in these headings. Now first let us discuss this genetic variation. This we will discuss first. Now the genetic variation, that means how the variations take place in different organisms, that is also discussed in different points. Because for variation, it takes place in different ways. Now number one is mutation. The second one is gene recombination. Then the third one we can tell that is gene migration. Which is also known as gene flow. Then the fourth one is genetic drift. Which is also known as Sewall-Wright effect. Then the fourth one, the next point. The next point is non-random mating. And the last point that we'll discuss is the hybridization. That means this due to 6 different regions, the genetic variation takes place. So we'll discuss this later on. Let us discuss this first, genetic variation. That means genetic variation is the raw material for evolution. But how this genetic variation is created? These are the different parts. Let us discuss one by one. So let us discuss first this mutation. So mutation means a sudden heritable change. Sudden heritable change is known as mutation. This already we have discussed during the Foucault-Davis theory of mutation. Now the mutation when takes place, it is again of two types. The mutation, it can be chromosomal mutation, or it may be gene mutation. Now the chromosomal mutation may be due to change in chromosome number, and this is due to change in chromosome structure. Then that we'll discuss. Now change in chromosome number, it is again two types. It may be polyploidy or it may be aneuploidy. Now the polyploidy is again of two types. It may be autopolyploidy or it may be allopolyploidy. Now all these things in detail we have discussed in the principles of innate. That means in genetics chapter we have discussed all these details. All these things, the change in structure, gene mutation, everything we have discussed. But here just I am giving only brief idea in 1 minute. Now when there is change in chromosome number, it may be in multiples. Multiples means say the single set may be n, then it may be 2n, 3n, 4n, 5n like this. Then it is polyploidy. But if there is change in individual chromosomes, one or two chromosomes are changed, that means that is known as aneuploidy. Again in autoploidy, it may be triploid, tetraploid, pentaploid, hexaploid like that. That means if the same genome is present in 3 sets, it is autotriploid. If they are present in 4, that is autotetraploid. But here there is no addition of new genes, only the existing genes are multiplied. So the variations are less. But in case of allopolyploidy, since there is a hybridization and then increased chromosome number, that is why this produce new alleles, they produce new characters, and that plays a very important role. Suppose one plant is say AA, the other is BB. Then the hybrid will be AB, which is sterile. Now if there will be, its chromosome number will be multiplied, then it will be ABB, which is fertile. Now it is a allotetraploid. That means here the character of both A and B are mixed together, and this plant is completely different from either of the two parents. So by this method, what we have obtained? We have obtained the triticum aestivum. We have obtained by crushing the different plants that we have already discussed. Then the second example is triticle. That is by crushing the triticum aestivum with the sickle cereal. Another example is Raphanobrassica. That means a cross between Raphanus sativus, that is radish, and Brassica oleracea, cabbage. So these are how the new species have been evolved. That plays very important role in the evolution. But coming to aneuploidy, there is change only in 1 or 2 chromosomes. It may be 2n plus 1 trisomy, 2n plus 2 tetrasomy, 2n minus 1 monosomy, 2n minus 2, then it is the monosomy, and it is disomy. So, or we can tell it is nullisomy, monosomy and nullisomy. So, trisomy, tetrasomy, monosomy and nullisomy. So, by this also, there is allelic changes. So, this is about this polyploidy. Now, when there is change in chromosome structure, the changes may be, number 1, there may be deficiency or deletion. Number 2, there may be duplication of genes. There may be inversion, that means a portion of the chromosome will be inverted, or there may be translocation when there is exchange of segments between two non-homologous chromosomes. So all the details again we have discussed in the principles of inheritance, that is genetic chapter. Now come to gene mutation. Gene mutation means the mutation takes place in the base sequence of a gene, and since it takes place at a particular point, the gene mutation is also known as point mutation. Point mutation. Now the gene mutation or point mutation, the details we have discussed in the molecular basis of inheritance. Again also we have discussed in the genetics chapter. So there is no question of repeating here, but briefly I am telling that the gene mutation, it can be due to frame shift mutation and substitution mutation. And the frame shift mutation, it may be due to deletion or due to addition. For details you can refer the videos of genetics. So this is known as point, and the agents which induce mutation, they are known as mutagens. Mutagens. All the details we have discussed earlier. So due to these mutations, maybe chromosomal or gene mutation, there is changes in the alleles of the organism, and that leads to evolution. Now let us come to, so the first point is over. Now come to the second point, that is gene recombination. So gene recombination, that means there will be reshuffling of genes. So when there will be reshuffling of genes, naturally the first point will be there must be dual parentage. Now we'll discuss the second point, that is gene recombination. Gene recombination. So what are the different reasons for this gene recombination? Now one may be due to dual parentage. When offsprings are produced sexually, two parents are required. So that is one of the important points. The second important point is independent assortment. Independent assortment. And regarding the independent assortment, again in detail we have discussed in genetics and also in the cell cycle. Where when in metaphase 1, the chromosomes orient themselves. Depending upon the orientation, either the dominant allele or the recessive allele will go to one pole, and accordingly the gametes will be produced. So that is also responsible. The third point is crossing over. Crossing over during meiosis. So as again you know that during meiosis there is exchange of segments between the non-sister chromatids of the homologous chromosomes. That means there is exchange of segments. So naturally the characters vary. So that is one of the reasons. Then the fourth point is a random fusion between gametes. Random fusion between gametes. That means the different gametes which are formed, they are completely different, because either the crossing over has taken place, independent assortment of genes have taken place. So the gametes, they are haploid number, and they may contain any number of any alleles, either recessive or dominant. So depending upon with which the male one will combine with which female one, so naturally the zygote will be completely different. So this is one of the main reasons. Then another point is also we can tell that formation of new alleles. formation of new alleles. Now, usually we know that each gene has two alleles, but due to some reason, if there will be more alleles will form, as you know, in the case of human blood, there are three different alleles. So, if there will be more alleles, that will add to variation, which will lead to evolution. So, these are some of the reasons under gene recombination which add new alleles to the population. New alleles to the population. So now the second point is over. Now come to the third point. The third point is gene migration, or we can call it also gene flow. Gene migration or gene flow. Gene migration or gene flow. Now suppose a particular population from one place will migrate to another place, and the new place of their settlement they will breed with the original host population. So what will happen? Now here the new alleles will be formed because a population from a different which have immigrated to a new population, and now the new population is breeding with the host population. So naturally new characters, new alleles will be formed, and that will create a variation which will lead to evolution. So that is known as the gene migration or gene flow. So this is usually, that is a movement of certain population, movement of population from one place to another place. This is one point. Second, then next point is breeding with host population. Breeding with host population. So when they will breed with host population, that means the characters, the alleles which are present from this in the migrants which have immigrated, now they will add new alleles, new traits to the host population. So when they will breed with host population, that new alleles are added. New alleles are added to the existing ones. Now, if let us take one example. Suppose in a pond, there is a dark colored frogs are there. In another pond, there is a light colored frogs are there. Now suppose the dark colored frogs will migrate to the other pond and they will breed with the light colored ones. That means the dark colored frogs will migrate from one pond to another pond where there is a light colored frogs, and they will breed with them. Now when the offspring will be formed, they will have completely different alleles from the original population, and the genotype, the genetic makeup of the host population will now change. There will be addition of new alleles which will create a variation, and that will lead to evolution. So this is known as gene migration or gene flow. Then the fourth point is genetic drift.